Hi everyone, welcome to the Bio Edge.
The following are some musings on the matter of energy deployment in Hemiptera. Sap-sucking Hemiptera, which occur in a bewildering diversity of suborders, superfamilies, families, genera, and species in the Hemiptera. They all have one thing in common, and that is that they tap into the juices of the plants that they live on, and they have to get rid of or otherwise deploy the excess sugar in their food. There's excess sugar in their food because phloem sap in particular, which is the main food of sap-sucking hemipterans, is essentially a dilute sugary solution with an even more dilute solution of various nutrients such as nitrogen, phosphorus, zinc, and so on. And to build their little bodies, these insects, such as aphids, for example, but including many other types, have no real direct metabolic use for all that sugar relative to the nutrients that constitute the building blocks of their cells. As their cells divide and they grow, they have a hopelessly skewed ratio in favor of sugar to the building block nutrients, such as protein, but also the various mineral nutrients that constitute the active ingredients in enzymes, the stuff of life. And so all of these sap-sucking hemipterans, they essentially use the superfluous sugar for some purpose beyond merely just excreting it. Some of them do merely excrete it, but in the form of honeydew, which is a dilute sugary substance that can be thought of as urine. But even that's a little complicated because simply peeing out large quantities of this sugary urine brings the risk of attracting sooty mold and other fungal infections, as it were, that can contaminate the colony of sap-sucking insects, usually nymphs. Bearing in mind that all hemipterans have incomplete metamorphosis, hemimetamorphosis, and they go through a series of instars called nymphs before finally graduating to the often winged adults. Not always winged, but usually winged adults, particularly in males. So the point is that we have a fundamental biological pattern that is kind of disguised in the bewildering diversity, taxonomic, phylogenetic, life history, strategy, and other forms of diversity in this bewilderingly complex group of creatures. There's both an overwhelming commonality and an overwhelming bewildering diversity. The commonality is that they all have to do something with all this superfluous sugar in the sap that they suck. The diversity is in the ways that they do this. And as far as I know, although there's an enormous literature on sap-sucking hemipterans, nobody has ever really provided a succinct account of the range of strategies used by sap-sucking hemiptera in deploying. I'm not sure if the word deploying is right, but I'll use it anyway. In deploying all this excess superfluous sugar for some kind of benefit, optimizing the benefit to cost ratio, as it were, in the way that all animals do, all plants do in their own way, in order to carve out a niche in the competitive biological world. So essentially what some hemipterans do is they simply ooze out the sugar as something that you might call urine, although it's not always crystal clear that it is excretion as opposed to secretion that is happening. But if you put aside that ambivalent distinction between secretion and excretion, this kind of rather simple-minded sap-sucking hemipteran, of which many aphids are an example, basically ooze out droplets of dilute sugary liquid that then either simply get dissipated in the environment by rain, for example, in which case it qualifies as simply excretion, even though it could be glands that are doing it as opposed to the alimentary canal. But in many cases, this sugar is used by ants in particular. And the ants then have the potential of entering into a mutualistic relationship called a trophobiosis with the sap-sucking bugs, in which the sap-sucking bugs, in their own passive way, supply a primary source of energy, namely sugar, to the ant colony, which in turn treats the sap-sucking buds somewhat like we treat cattle, and manages and husbands them, and certainly protects them from their own predators, which are legion. And in this way, you have a useful mutualistic relationship based on the consumption of the sugar by ants. So that's kind of the baseline and the best known deployment of superfluous sugar by sap-sucking hemipterans. And this kind of trophobiosis between the hemipterans and the ants occurs in many different clades of Hemiptera, which we don't need to go into in any detail for now. The second possible use is in a kind of an investment in which the sugar is converted to something structural that can then protect the vulnerable little sap-sucking nymph. Bearing in mind that the whole foraging strategy of Hemiptera is to be kind of stuck in to the plants that they suck the sap of. Once you insert your complex microscopic proboscis into the phloem of the plant that you're sucking the sap of, it makes you vulnerable because you can't instantly just pull out that proboscis and move off rapidly. And indeed, most of the nymphs of Hemiptera are not capable of rapid movement at all. They're rather sedentary creatures, the lives of which depend on this kind of rooting, almost, the rooting of the mouth in the substrate plant. And because that makes them vulnerable to being picked off by gleaning birds or indeed ants themselves, what they do is they use the superfluous sugar to produce a kind of a shell around themselves. But the word shell is hopelessly simplistic in describing the unbelievably elaborate 3D structures that are involved in some cases. Now, the simplest case in terms of biochemistry, although not the simplest case in terms of structure, is a kind of a shell called LERP, L-E-R-P, which represents a polymerization of the sugar into a polysaccharide. So it's quite a simple chemical conversion. You just take the sugar and you make it into a starch, mainly a starch. And these mainly starch structures have the advantage that they're not soluble in rain, and they gain a certain measure, a modest measure of durability, which means that the whole nymphal series in the growth stages of the Hemipteran, in this case certain genera within the Psyllidae family, the jumping plant lice family, which is in turn in the superfamily Psylloidea of the suborder Sternorhyncha of the order Hemiptera. In this case, what you have is a sort of a starchy shell that then effectively becomes part of the body of the nymph concerned. It may as well be part of the body of the nymph concerned, even if there does not remain a direct connection between the starchy exudate or secretion and the actual glands, complex glands on the abdomen that produce that, because the animal is so sedentary that if its LERP shell is removed, it basically dies because it does not have time to secrete a new shell, and even just exposure to the weather and to the sun and to the elements is going to knock it off. So the LERP shell, the starchy shell, and the tiny little nymph hidden under it are essentially one body, one flesh, as it were, even though the LERP substance itself is not flesh. It is a secretion which was never alive any more than the oil that comes out of the sebaceous glands in your scalp was ever alive. So at this point, just a little digression to dwell on the subtleties of this. Your hairs as a mammal were alive because the follicles, the hair follicles on your scalp produce a living tissue at the base of the hair, that then dies out and dries out to form your hair, which is essentially a plastic form of a tissue that still contains the original DNA, because it's a whole bunch of cells that have become plasticized. The starchy lip of a hemipteran is quite different, because it is not a tissue gone indurated. It is a secretion, a little bit like a 3D printing. There are fairly complex glands on the abdomen of the nymphs of these psyllid hemipterans, and these glands 3D print through the process of complex secretion what amounts to a fascinatingly complex and relatively durable starchy shell-like encasement, which we call lerp. But we may as well use the word lerp to refer to the combination of the shell and the nymph that produces it and hides under it, because to all intents and purposes, in the real world, they are one flesh. And this raises a very interesting phenomenon to light that has not been done justice to in the literature on hemipterans, and that is that there's a whole phenomenon in Hemiptera, which I call a secromorphosis, in which the body is greatly extended by secretion in a way that does not happen, as far as I know, in any other group of mammals. Right, so that's stage number 2. So just to review so far, the superfluous energy can be simply exuded. I use the word exude to avoid the having to choose between excrete and secrete. It's exuded in the form of sugary fluid, which can then be used to protect the animals by virtue of the trophobiosis with ants. Or it can be polymerized in a simple way into the polysaccharide that we call starch, which then potentially emancipates the hemipteran from needing ants, because it gives its own physical protection along the same lines as a tortoise's shell. It allows this little sedentary and plugged-in organism to have some measure of protection from the outside world. Okay, so you can see here that the psyllid nymph has done something biochemically to the sugar within its own cells by converting the sugar into starch, and then it has exuded it to produce this 3D printed shell. So that's a perfect example of having used the superfluous sugar for some beneficial purpose. Of course, there are costs, as in all things biological, to this strategy, but we don't need to go into them. It's self-evident, it's axiomatic that there's costs in every choice in adaptation. Now, the third stage is to do something more complex biochemically with the sugar feedstock, and that is within the cells of the nymph, and in some cases also within the cells of the adult of the hemipteran insect, depending on the clade involved. The enzymatic process is masterful enough that it takes the sugar that it has extracted from the sap, and it actually creates wax out of it. Now, this is not polymerization, because wax is not a polymer. It is a much more complex substance, and it's also complex in the sense that some waxes, like sugars, contain carbon, hydrogen, and oxygen, whereas other waxes are actually just hydrocarbon. They just contain carbon and hydrogen without the oxygen, which further removes them from the biochemical nature of the living material that generates them. So a wax is a strange thing for an organism to produce in the first place, because there's nothing metabolic about it. There are no waxes inside the cells of organisms. A wax is something that is produced by life, but is somewhat foreign to life, because it can only exist as an exudate, not as a part of the protoplasm. And when that wax is hydrocarbon wax, consisting only of carbon and hydrogen, it really gets into the realm of non-living matter, because most hydrocarbons in the world are not associated with life, at least directly. They are what we call, petroleum fuels, as it were, and they're also petroleum waxes. So in the sense, to the degree that the hemipteran bodies make waxes, you have kind of two stages in this progression that I'm sketching out in how sugar is deployed from simple to complex. Because in addition to the honeydew phenomenon, the first one, and then the starch phenomenon, the second one that we're calling lerp, there is a third one which consists of what we might call organic waxes containing carbon, hydrogen, and oxygen, which are complex organic molecules. They're not merely polymers of sugars in the way starch is. They are complex organic molecules, and their advantage is that they're much more recalcitrant in the sense of being difficult to digest by other organisms, and so they form particularly durable and useful shells and shelters for the hemipterans that secrete them. And then the fourth stage is the waxes that include a hydrocarbon component, which are even more valuable in terms of protection, because hydrocarbons are potentially not just indigestible to other animals, such as the predators of hemipteran sap-suckers, they're also potentially toxic. If you as a human use an organic oil, such as coconut oil, in your food, that's one thing. However, if you try and put engine oil, clean engine oil out of the can, out of the bottle, into your food, you're likely to be poisoned, because hydrocarbon oil is not the same thing as organic oil in your diet, and it's the same generally across the board. Hydrocarbons, where they are produced by various plants and insects, are somewhat inimical to being consumed, and so a hydrocarbon-based shell on a sap-sucking insect, the shell having been produced by secretion as part of the phenomenon of secromorphosis, this hydrocarbon-based shell is a step, again, the fourth step in our series of the complexity and advancement and sophistication of the deployment of superfluous sugar by sap-sucking hemipterans. So that'll do for now. I can probably add to this in a future segment. I don't want to get too long and complex, but the basic idea here is that all sap-sucking hemipterans have sugar to burn, as it were. They can't burn it in their metabolic process. They have no use for it there, and so what they do is they use it in a remarkably inventive, imaginative, and diverse way to offset their risks, to perform trade-offs, and to engage in a kind of a benefit-cost analysis that manifests in a wonderful diversity of not only life history strategies in the Hemiptera, but also in actual growth forms or life forms. Because when you look at, for example, a coccid sap-sucking hemipteran that belongs to the superfamily Coxoidea and the family Coccidae, what you see is a macroscopic, rather round, quite prominent lump of wax, the various individuals being aggregated. So there's a whole series of wax lumps on the surface of the plant in question, and under each wax lump you have a little nymphal coccid sucking away at the sap, having produced this quite substantial part of its body, for which there's no particular name. They call them scales. You can call them hard scales. Let's just call them hard scales. Okay, so the hard scale of an individual nymph of a coccid sucking away at the sap of a plant can be many-fold the actual body of the nymph. I suspect it's up to several hundred-fold, because the nymph is quite small, the wax hard scale is quite big, and so you've got a remarkable situation where most of what is effectively the body of this particular individual insect consists of a material derived from the sugar but converted enzymatically in the cells of the hemipteran, and then extruded, secreted by complex 3D printing glands on its abdomen. into a structure that is perhaps up to 95% or more of the body of the organism, but is remarkably in a category where it was never living in the first place. The moment it was extruded from the body of the nymph, it was already in the non-living category. So you have non-living matter produced by living matter, and then kind of remaining attached to the producer in the form of a shell, a wax shell, and sometimes a hydrocarbon wax shell that is powerfully protective to the organism concerned. And then to give you another aspect of this, there is another use for the sugar that we can almost take for granted because we're talking about insects here. And so I won't call this an additional stage in this progression because it's on a kind of a different axis, as it were. But it's worth mentioning that chitin, which is the main structural material used in the exoskeletons of all insects, including hemipterans, is itself a polysaccharide. So chitin, like starch, is a polysaccharide. It's not like keratin, which is the protein-based plasticky material used in hair and feathers and reptilian scales. Chitin, which is found in invertebrates, arthropods, is itself a complex form of sugar in the form of a polysaccharide that is, again, durable. It's a bit like, if you like, it's a bit like paper. Insects do not produce cellulose in the way that plants do. Instead of producing cellulose, insects produce chitin. But both cellulose and chitin are, in a sense, forms of paper. The cellulose is plant paper. The chitin is insect paper. But the point I'd like to make here is that, again, the capacity to make chitin is affected by the supply of sugar, because chitin, the main structural reinforcement in the exoskeletons of all insects, is a polymer of sugar. And this matters because within the Hemiptera in general, there's a kind of a gradient from certain forms, typically aphids, that have very soft bodies. The exoskeleton is minimal. And even in the case of the wings, in winged forms of aphids, the wings are minimally reinforced. They're just little perishable membranes that last for a few days and then get frayed and destroyed. Whereas other hemipterans, particularly in the suborder Orcanorhyncha, are heavily reinforced with exoskeletons and have rather large, hard wings. Not like the elytra of beetles, but they have rather hard wings that are fairly well reinforced by means of the same material that's used for the exoskeleton. And so, for example, frog hoppers are relatively well armored hemipteran insects, at least in the adult stage, by virtue of an exoskeleton. And there's this kind of trade-off within the Hemiptera in which those species, those clades that minimize the investment of sugar into the chitinous exoskeleton, are also those that are most generous in their deployment of that same sugar for other methods. Like in the case of aphids, the aphids minimize the exoskeleton and even render their wings to be minimally constructed. And then they take that sugar that other insects might have used to make chitin, and they actually just pee it out, as it were, in the form of honeydew. Others, again, like the coccids, they have a minimal exoskeleton, because if you pry off that hard scale from the surface of the plant, you'll find a very vulnerable, soft-bodied little nymph under it. And so instead of reinforcing the actual somatic or cell-based part of the body with an exoskeleton, the coccid nymph devotes all that sugar to the secretion of wax, which then substitutes for an exoskeleton. You see, this is the point, is that there's this gradient of strategies among the Hemiptera in which some minimize the exoskeleton and maximize the use of sugar for other things in the form of, say, honeydew. And then there are others that honeydew or a wax scale, whereas there are other hemipterans that may produce some honeydew and some wax, but reserve the right to channel quite a lot of their superfluous sugar into making a substantial exoskeleton for themselves. And that exoskeleton, again, is relevant to this discussion because it is made of a sugar polymer, a polysaccharide called chitin. And perhaps the extreme example of this is certain orchinorhynchine hemipterans, particularly in the family Membracidae, which have such an impressive exoskeleton in which the chitin has been packed into the exoskeleton to such a degree that these insects in their adult form actually resemble spines, thorns, like plant thorns. And they actually defend the adults from, for example, insect-eating birds by being so spiky that they function as thorns. And so they both mimic thorns to disguise the insects as they perch, often lined up on the stem of the plant, and or they actually defend the bodies of the adult hemipterans as thorns would on a spiny animal, albeit only one or two thorns per individual insect, but enough to make the object difficult and perhaps even injurious to swallow by an insect-eating animal. And so, as you can see, that's another theme in the Hemiptera. So you have Hemiptera that are strongly defended by an exoskeleton, and you've got Hemiptera that are minimally defended by an exoskeleton. And that corresponds with the sugar being used in inverse correlation. And so the waxiest and sugariest colonies of sap-sucking hemipterans are likely to be the ones with minimal exoskeletons. Whereas the converse is true. For example, in membracids, the membracids do produce some honeydew and they do produce some wax. But a lot of the sugar that is superfluous to the metabolic processes within the cells of the insect is then deployed into exoskeleton in a way that the exoskeleton actually exceeds what you would expect for an insect that's trying to protect itself out in the open environment. And so this whole topic is full of, shall we say, gradations in a particular pattern of deployment. And it all revolves around the common theme that every sap-sucking hemipteran in the myriad clades of sap-sucking hemipterans has the same basic advantage as well as disadvantage. Every advantage is a disadvantage. Every opportunity is a crisis. Every crisis is an opportunity. And the basic fact about Hemiptera, upon which this whole voice clip depends, is that if you are a sap-sucking insect, you will have superfluous sugar. What you do with it then is your business, and one of the reasons why entomology and biology are so endlessly fascinating is that the imagination of the evolutionary process seems to know no bounds, and it comes up with all these different, almost capricious approaches to how the superfluous sugar can be used that then have consistent correlates. Because if you take one tactic, then that means through the principle of opportunity costs that you don't really have the option of other tactics. And so it's this wide variety of trade-offs within the Hemiptera, all, shall we say, centering on the fundamental phenomenon of a superfluity of sugar that I think is worth knowing about, even if you're not particularly interested in entomology, even if you are wary of getting into the weeds with hemipteran taxonomy, which is indeed bewilderingly complex, even if you know nothing else and want to know nothing else about the biology of Hemiptera. I think this one principle, this pattern, this, shall we say, syndrome is worth knowing about. And that is that there's sugar available, let's do something with it. And much of the phylogenetic diversity of Hemiptera, all these different clades, is based upon the different choices made in how to use the superfluous sugar. And with that, like, subscribe, and share. And we look forward to seeing you next time for our next segment here at the BioEdge. And over and out for now.